It's my honor to introduce our first plenary speaker today, who's Nim Tottenham, for whom most of you needs absolutely no introduction, but I'm going to give a brief introduction anyway. Nim is Professor of Psychology at Columbia University and Director of the Developmental Affective Neuroscience Laboratory. Now, she is a Fellow of the Association for Psychological Science and was one of the early recipients of the highly prestigious BRAINS Award. That's the NIMH Biobehavioral Research Awards for innovative new scientists. And I think those of us who know Nim knows that, she epitomizes the BRAINS Award in terms of her innovation and her ability to grasp so many ideas and put them into practice in the developmental space. I think I have to say, even though it's not on her website, and I was just talking with Nim earlier, most of us probably know Nim for the Nim Stim, the facial stimuli package, and I know my lab uses her stimuli all the time. I also want to note that Nim has done fantastic research in the developmental space. She examines brain development, underlying emotional behavior, and really is really focusing on the powerful role that early experiences, including caregiving and stress, actually help shape the normal human development. So without further ado, I want to introduce Nim, and she's going to be talking about the development of emotion regulation, neurobiology, and the fundamental role of early experiences. So please welcome Nim.
Thank you so much. Thank you. Good morning. I'd first like to thank Mary and Scott and the whole staff of SOBP for organizing this amazing meeting. So the work in our lab addresses this question, which many of you do, trying to understand the links between early caregiving experiences and emotional functioning later on in life. And for this talk, I'm going to be focusing on the bidirectional connections between the amygdala and the medial prefrontal cortex. Of course, many more regions of the brain are responsible for emotion regulation behavior, but we're focusing on this set of connections because in adulthood, this really forms the foundation of what we consider responsible for emotion regulation behaviors. And our approach is to understand how this system is constructing itself during development. Now, if we look at a number of mental health problems associated with affect and affect regulation, they seem to emerge around the beginning of adolescence. Estimates are that a large majority of these are attributable from early adversity exposures, and as the authors acknowledge, this is a gross underestimation. So given that, our approach is to look at the antecedent period, what's going on when the system is constructing itself and how that construction is interfacing with that children have. So in his review of the literature, Amit Ekin highlighted that connections between the amygdala and medial prefrontal cortex in adulthood often are anti-correlated with each other. You get this negative correlation between amygdala and medial prefrontal cortex, the hallmark of emotion regulation in healthy adults. So we interrogated this developmentally, and we and many other papers I'd like to highlight here have identified large changes in amygdala PFC circuitry. So what I'm plotting here is age on the x-axis and amygdala reactivity to things like fear faces. And so each dot here is a participant, and what you can see is the younger participants are showing this very strong amygdala response to fear faces. And that makes a good bit of sense because the amygdala has a central role in learning about the relative safety and danger of the environment, and when you're young and new to this planet, you have a lot more learning to do. We can then use functional connectivity during task and assess the nature of these changes in connectivity. And we found, we replicated what many other labs have, that in young adults we get this nice anti-correlation between amygdala and prefrontal cortex, indicative of a regulatory pattern. But in children, we see something quite different. We don't get that anti-correlation, but instead amygdala PFC are kind of moving together, suggesting that this is a non-regulatory pattern. Now, that switch in connectivity valence has been associated with declines in normative developmental anxiety, like separation anxiety. So putting these types of data together, if this is age on the x-axis, we see, at least in childhood, we're getting this strong amygdala response to emotional events, but that's happening in the absence of these adult-like connections with medial prefrontal cortex, which we don't see emerging until the beginning of adolescence. So that's made childhood a very interesting time for us to think about when the environment might have a particularly strong influence on the way that these connections develop over time. And in a twin study performed by Michelle Ochterberg in the lab of Evelina Krohn, they examined resting state functional connectivity in 7 to 9-year-old twins. And what they identified was that the connections between amygdala and medial prefrontal cortex seem best explained by environments as shown here. So no influence from genetic factors in these data. And the environment that we're particularly interested in is the species expected input of the caregiver. And humans have caregivers that can be their biological mothers, their fathers, grandparents, etc. This is a learned relationship, as it is in many mammals. But one thing perhaps to point out in the human is that, unlike other species, we spend a very long time hanging out with our parents. And that is metabolically very costly to a parent to raise another human being, so there's got to be a big payoff. And the explanation that we find very compelling is that the payoff is this luxuriously long period of childhood when we have exposure to many, many emotional experiences that give rise to the exquisite ways that we interact with emotional stimuli in our future. So we can think about parents as providing this scaffolding, like on a building, that helps guide development of the central nervous system during a time when it's yet immature. And so for many years, we've been very compelled by the rodent literature, in particular that of Regina Sullivan's, which has shown this intimate relationship between the parent and development of stress-responsive systems. So for those of you that are less aware of these data, there's this period in development when the functioning of the amygdala is critically dependent on whether the mother is in the nest or not. When the mother is in the nest, she leads to a neurohormonal cascade that dampens activity of the amygdala, and when she's out of the nest, those processes reverse, so amygdala can now engage in learning. And so in other words, the mother is turning on and off the engagement of the amygdala during learning. And to test that behaviorally, she uses Pavlovian fear conditioning, pairing a peppermint odor with a foot shock, and then places the peppermint odor in one arm of a maze, control on the other. If the animal conditions without the mother, it will do what you and I would do, which is avoid that peppermint odor, as shown in this small bar here. However, if the mother is present during conditioning, then this animal, despite the fact that it does not like the foot shock, will now approach that peppermint odor. If that sounds bizarre to you, it is in a way, but it matches the ecology of this animal at this developmental point. So in other words, survival strategies like fight or flight are not useful strategies if you can't do either well because you're very young. Instead, the survival strategy of the young altricial animal is attachment. That is to say, regardless of the quality of care you receive, whether warm or painful in this case, the brain is positioned to attach. So attachment learning will trump fear learning in the presence of the mother in the rodent. So Regina and I were very interested to ask whether something similar could happen in humans, especially given that human development is so drawn out compared to the rats. And so with about 100 preschoolers, we presented them with a blue square co-terminating not with a shock, but an aversive noise like loud nails down a chalkboard. The purple triangle was paired with nothing. They learned this in the presence of an experimenter or their parent who was just filling out paperwork, but they were in the room. After this, the parents left, and we put children into a human Y maze, and just like in the rat study, asked whether they showed differential preferences for the CS plus as a function of the learning environment. So first, I want to show you the acquisition data that occurred during this moment up here. There's a lot of data in here, but I will walk you through it. So ignore the differences between blue and gray right now. By 4 different metrics, we had a reaction time, accuracy, heart rate, and skin conductance during acquisition. 3 to 5-year-olds were able to discriminate CS plus from CS minus stimuli reliably. But if you pay attention now to the gray data, which is when they conditioned alone, versus the blue data when they were with their parent, there was no difference. So parents did not seem to be affecting acquisition data, at least at observable behavior. So when we then looked at their performance in the Y maze, now the parent is gone. When children conditioned alone, those stimuli were overall avoided by children. But when the children conditioned in the presence of their parent, when they were in the Y maze, they now showed a modest yet significant preference for that CS+, really paralleling what was shown in the rats. Whether it's the same exact mechanism, we can't say at this point. But I want to also remind you that this was a within subjects design, so these are the same children whose behavior, whose learning is changing as a function of who was in the room with them. The fact that the parents weren't affecting acquisition, but they were affecting learning, suggests to us that what parents are doing is they're changing the nature of what was learned and recalled later on. This effect was attenuated by higher court levels, which again, it's early days, but parallels the rodent data where developmental increases in court is the stop signal for the parent's effect. So parents can no longer have that effect after that rise. We also scan children while presenting them with pictures of their parent, contrasting that with pictures of somebody else's parent. And here now children are showing a decrease in amygdala reactivity as opposed to that very strong response I started the talk off with when looking at their parents. We're currently interpreting this as parental buffering, but we're doing a lot more work to better understand that. Adolescents don't show that effect, and that's not because adolescents aren't important during parents aren't important during adolescence, they certainly are, but maybe at the level of the amygdala they've started to lose their efficacy. Parents were also effective in momentarily or phasically strengthening that anti-correlation between amygdala and prefrontal cortex in children, and that effect was stronger for children with stronger attachments. So these types of data have made us think this way, that there's some period in childhood, maybe a sensitive period, when parents are routinely changing, modulating the circuitry, when parents' availability or unavailability are leading to different neurohormonal cascades. And the idea is that the nature of the way that this system works in adulthood is a reflection of the cooking that happened during childhood. So parents may be a privileged information source into limbic circuitry during childhood. So this framework allows us to then ask questions about aberrations in caregiving, and we've been particularly interested in neglect, and we've been asking this question for the past 2 decades, studying children who experienced early institutional care followed by adoption. And just to state the obvious, we think about omission in these instances of neglect, but omission of input that's expected biologically is not the same thing as the absence of harm. So it's the absence of an input, but it's a highly pernicious experience, and unlike abuse, where the signs are more obvious, neglect effects are often invisible and thus often overlooked, but very common and cut across other types of maltreatment. It's the failure to receive needed parent-child intimacy, support, serve, and return, and children are often described as raising themselves. And this is a significant stressor during brain development. So if we think about this as a stressor, we can turn to decades of research on stress, and these are adult male rodents showing that chronic stress leads to hypertrophy and hyperactivity of the amygdala, and the opposite is true in the hippocampus. And I like contrasting these two because it reminds me that stress isn't having a uniform effect on the brain, but it's really informing the brain of how to better respond to future stressors. Now, the children that we see, even in the best case scenario with the best of intentions, have experienced a major stressor and are therefore at great risk for developmental delays. There's significant rebound upon adoption, and that's perhaps not surprising because the families that adopt, these are international adoptions, tend to, we infer, have a great desire to provide care because it's not easy to adopt internationally. There's tremendous heterogeneity, as I'll show you. Some children are really thriving and some are struggling. And when they're struggling, it's often in the domain of dysregulated affect or behavior. So if we look broadly at things like internalizing and externalizing, you can see that there's a large effect size of previous institutional care on both. But I also want to highlight, these are data in 373 children, the individual differences. And so a lot of our work is targeting how do we better understand the differences between this child up here and this child down here. So we used a face go no go task developed by BJ Casey, and it's a great task because children can do it, and they're told to press a button as quickly as they can every time they see one facial expression, but to inhibit, which is very hard for them to do when it's a different facial expression. And we can change the valence of this face to see how emotional valence impacts performance. For positive emotions, we didn't see any group differences. The group differences really emerged for the negative faces, and that's highly consistent consistent with parent clinical report that children tend to have greater dysregulation problems in highly arousing situations. And during scanning this task, both in New York and Los Angeles, we identified hyperactivity of the amygdala within this group. Now, when we started reading the rodent literature, many studies were showing that early life stress, as opposed to later life stress, may have a different immediate phenotype, which is to accelerate development of stress-related circuits. And so I list here some of those examples: premature myelination, BLA learning, fear reinstatement, and so on. And the idea behind all of these papers is that when identifying differences during development, what we might actually be seeing is evidence of developmental adaptations, not phylogenetic, but developmental adaptations, which are activity-based changes in brain development to meet my immediate needs. And if we consider these as adaptations, it allows us to better think about trade-offs between benefits and costs. And so we looked at connectivity between the amygdala and PFC, and these are the data I showed you earlier, where typically raised children show this childlike phenotype that switches to this anti-correlated regulated phenotype in adolescence. We replicated that in a separate cohort. Following previous institutional care, we do not see that childlike pattern, but instead we see what looks more like an adult pattern. Whether this is a true acceleration or not, we can't say yet, but these data suggest that this is an adaptation for reasons that I'll explain. So the association between early life stress and connectivity was mediated by cortisol, which again is a very nice parallel to what's been shown in rodents, for example, in Rick Richardson's work. And so our thinking is that the hyperactivity of the amygdala as a consequence of this early stress may actually be the biological prerequisite for instantiating those adult-like connections with prefrontal cortex earlier, and that may help an individual. However, at the same time, that may also truncate or reduce that period of developmental plasticity that we think is so important for mature emotion regulation. So to further inquire about this potential trade-off, we looked at behavior, we looked at anxiety and separation anxiety here, current separation anxiety, and we find that following previous institutional care, children are showing more anxiety, but if they show the more adult-like phenotype, which about 60% of our sample did, then they seem to benefit. They had lower anxiety than their previously institutionalized peers who did not. And then we can look at the longitudinal data, and these numbers are smaller, so they require much more investigation. But if we follow these children over the next 5 years, we find that that rank order switches, so that those children who were showing the more adult-like phenotype and were benefiting in terms of anxiety did not show the same declines in anxiety over time and tended to hang on to that phenotype, whereas the children who showed the more childlike phenotype and were struggling more with anxiety had steeper declines over time. And so this raises the very interesting question about transactional effects in this group. If the children the children who are showing more separation anxiety may be eliciting different caregiving behaviors from their parent that in the long run benefits them. So finally, we asked if acceleration was abbreviating plasticity, would we see limits on this parental buffering of the amygdala? So these are the data I showed you earlier, where children with a typical caregiving history showed this dampening of the amygdala response in response to their parent. Following early institutional care, we did not see evidence of that same buffering, suggesting that at the group level, there were limits placed on this buffering. However, we were paying attention to, as I'm sure you are, these very large error bars here, which suggest that some children, even following early institutional care, were showing buffering and some were not. So we interrogated those individual differences longitudinally and observed that those children who were showing buffering of the amygdala at time 1 by their parent had steeper declines in anxiety than those who did not show buffering at time 1, despite the fact that their levels of anxiety were the same at time 1. And what predicted those individual differences was not age of adoption or the usual variables we would look at, but the child's reported attachment security. So children who did show buffering to their adoptive parent at time 1 also reported more secure attachments at time 1. And that is consistent with other data we've collected showing that following early institutional care, higher attachment security was also correlated with decreased internalizing problems. So these data suggest that attachment is always important, but it may be especially powerful in ameliorating some of the adverse effects of early adversity for those children. So to conclude, I've said that human cortical subcortical circuitry develops very slowly, but perhaps there's value to this slow cooking. This gives us a long sensitive period for the environment to influence amygdala medial PFC connections in childhood. And therefore, parents can provide emotional scaffolding for children's brain development. Early caregiving adversity does significantly increase the risk for poor mental health, but there are large individual differences, and we're only at the earliest days of interrogating those individual differences. We think strong families are a powerful source of change, especially for children who experience early adversity, and that's an area we think requires much more work. And finally, we've suggested that qualitative differences in outcomes as a function of early life stress may actually result from changing developmental pacing. And so we think that by understanding these phenotypes, some of them as developmental adaptations, may get us a new way to think about mechanisms of change following early adversity and improvements. So with that, I would like to thank the funding sources, lab members past and present, families and individuals who participated, and thank you for your attention.
We have some time for some questions.
Terrific work. Thanks very much for this presentation. Did you find any sex differences in those results that you presented?
Yeah, so it's too early. We're underpowered yet. We're doing a much larger study right now to address that question. The one place where sex differences came up was not in the adversity data that I showed, but in the human Y maze data. It wasn't anticipated. But to make a long story short, girls, as they got older, it was just 3 to 5 window, but as they got older, they started to lose that influence of the parent, whereas boys tended to retain that influence of the parent throughout all of the ages, which, if girls' emotional development is a little bit more quickly paced, might be an indication that there's a window that girls are exiting out of earlier than boys.
Hey, good morning, Nim. Great talk, as usual. I had a question regarding the parental attachment or the relationship attachment moderating effects you're seeing, both on amygdala connectivity development as well as anxiety symptoms. Do you see also any effects of parenting style? So the amount of warmth, for example, or harshness, does that influence these trajectories as well?
Yeah, that's a great question, and it's something we're very interested in. We do have those data, and we haven't looked at them yet because our approach has been to use this blunt instrument of parent there, parent not there, because we think that that's important. But certainly the next step is to start looking at actual behaviors, because it could be that the usual suspects emerge, and it could be that it works differently following early adversity, because children are eliciting different things.
Yeah, sure.
Thank you. Yes.
Thank you for the great talk. I would like to ask about the sensitive periods, because your data is showing that there is a sensitive period in childhood, but not only during the first years of life, but also extending to later, like to 3 to 5, maybe to even to 10 years of first years of life. But still we have a common understanding that the first years of life would be especially important due to the rapid neurodevelopment. So what do you think from the perspective of your data, like about that is there anything specific about on the first years of life or the early postnatal period, or is it something in terms of parenting that seems to be taking place throughout the childhood?
That's a great question. So, I would refer you to a lot of the writings of people like Chuck Nelson who've talked about these kind of hierarchically organized sensitive periods depending on which brain regions you're talking about. And so, given the hierarchical organization of the brain, we would predict that the first year is really, really important. But then so are the subsequent years because of the things that are building on snowballing on top of that early experience. But then it raises the question of interventions. And so if interventions are working, are they going to have to be sort of made unique towards the age at which some insult occurred?
Thank you so much.
Thank you.
Do you have time for another quick question?
Yes.
Okay, thanks. I was just wondering around the increase in anxiety that you saw with those who had the adult type pattern, and whether another possible explanation other than the one you offered, that maybe those who had somehow higher separation anxiety had elicited greater caregiving response, might be the reverse, that those with the adult type profile had taken on an adult type role at a too early stage, which then leads to enhanced anxiety. I wonder if maybe they were taking an adult type role amongst their peers in the institution.
Thank you for your question. I think that's a really important way to think about it as well, and it reminds me of decades of research that have looked at things like early parentification of children who experience stress. And while those behaviors are, on the one hand, good, children look responsible, feeding their siblings breakfast in the morning and so on, because there's not a parent who can do it. Those are the same individuals that are also at great risk for internalizing problems later on. And so I think I'm not sure that that explanation is entirely different than thinking about what the kids with high separation anxiety are doing. But I think that it fits in this framework of trade-offs, that there are some advantages, but there might be some longer-term costs.
Sure. Thank you very much.
Thank you.
Very nice work.
And I'll take my prerogative to ask a question. I've loved your work for a long time. It's outstanding. So we often get asked the question, how does this lead to intervention? And I think you teased us with that. So how would we actually change the way we intervene with these kids?
So I'm not a clinician, but the data that I've seen both in humans and in rodents really just draw me to the family. And even I've spoken with many caseworkers over the years, and the answer is always the same, that it's about the family. And so really thinking about how to not only treat the child, but, to quote Winnicott, there is no such thing as an infant. It's really treating the family and finding supports for the family. Now, the data that I didn't show was that anxious or distressed parents are equally as good at potentiating all of these fear systems. And in Regina Sullivan's data, again, that potentiation can occur at an age when the amygdala isn't even supposed to come online yet. So, parents really are moving the dial around a lot, and so I think that paying attention to the health of the family is an important next step.
All right, great. Thank you so much.
Thank you.